A good day and a very warm welcome to Dr. Ayub and my fellow friends. We are from Group 3D. I'll start off by introducing myself. My name is Navinish. The title for our experiment is The Activated Carbon Adsorption Isotherm and the Kinetics Involved in the Removal of Methylene Blue from Wastewater in Textile Industry. Our experiment includes the application of the Experiment 9. The reason why we chose textile industry is because textile industries have been the epicenter of worldwide water pollution problem. Dyes and pigments which involve direct, reactive, acid, and basic reactions are widely used in the textile industries, particularly for coloring the fabrics. As a cationic dye, methylene blue is commonly used for dyeing cotton, silks, paper, plastics, or even leather. It is very important to remove such dyes from wastewater before discharging them into rivers or ocean. There are two main objectives in this experiment. The first objective is to study the effect of contact time, pH, and mass of adsorbent on the percentage of removal of methylene blue by using activated carbon. The second objective is to determine the best fit adsorption isotherm for the adsorption process by plotting Langmuir and Freundlich isotherm.
Hi, I'm Masdena. I will present about market analysis. Methylene blue has a wide downstream application and it has recently gained prominence in industries such as aquaculture and pharmaceutical. The global methylene blue is primarily driven by the rising of aquaculture and pharmaceutical demand. According to the Global Methylene Blue Market 2019, methylene blue is consumed in about 62.54% of the world aquaculture and pharmaceutical industries. It's also reported that Europe is the largest consumption market with 30% of the global consumption of methylene blue in 2016. All the market type of methylene blue are expected to keep increasing from 2013 to 2024. Next, methylene blue consumption is expected to grow steadily in the coming years from 2019 to 2027. It is predicted that the market size of methylene blue to achieve 90 million USD in 2026 from 70 million USD in 2020. In 2021 to 2026, the compound annual growth, which called as CAGR, is expected to be at 5%. The market size of methylene blue is expected to further being expanded in 2027 to reach more than 90 million USD.
Good day everyone, my name is Lojia Cheng and I'll be presenting to you the process flow diagram of the plant. The first part of the process flow diagram is the textile industry factories. In these factories, the wastewater will be discharged and the discharged water will enter the equalization tank. The equalization tank will reduce the pH so that it is more favorable for the microbial growth in the aerobic process for the next stage. The next step will be the aerobic process. As mentioned, this section involves the microorganism for treating the effluent from the factory. It is said that using microorganism is actually more environmental friendly. Then it enters clarifiers. There are two clarifiers with both serves different functions. The first is for the settling of the solid particles via sedimentation, and it also serves for pH adjusting. The next one is for the coagulation and flocculation of solid particles. Next, the effluent will enter the adsorption and filtration tank. This tank was filled with activated carbon for absorbing the color from the dye and the odor. Our experiment focused mainly on this section. For your information, the activated carbon is used as it is cheaper absorbent for the process and it has high capacity for removing the dye. After this part, ozonation takes place. Ozonation removes the dye via the addition of the radical-inducing compound for the breaking down of the dye. The second last step is ultrafiltration. Ultrafiltration separates the dye from other small solutes from the water. In this section, the rejects from the ultrafiltration will be recycled to the equalization tank, while the permeate will be transported to the reverse osmosis unit. In the reverse osmosis unit, the water is extracted from the remaining components. Hence, the water will be the permeate and the reject will be sent to the treatment plant. Water will be sent to the storage tank for the reuse and recycle of the plant. That's all from the process treatment plant. Thank you very much and have a nice day.
Hi, I'm Xiaojia and I'll present the results and discussion of this project report. Prior to the experiment, it is important to develop a calibration curve in order to predict the concentration of unknown sample. Therefore, we obtained the standard calibration curve of methylene blue dye at wavelength of 664 nanometer. The y-axis of the graph is the adsorbance from the UV ray spectrophotometer, while the x-axis is the concentration of methylene blue. The accuracy of the graph was excellent, since it has a coefficient correlation of 1. Also, we observed that the percentage of adsorbance increased as the concentration increased. For the first part of our experiment, we will be investigating the effect of contact time on percentage removal of dye. From the results, we observed that percentage of removal of methylene blue increased with increasing contact time. At maximum contact time, final concentration of methylene blue is the lowest. During dye adsorption, it is important to know that methylene blue molecule must first reach the boundary layer, then diffuse through the adsorbent surface, and eventually diffuse into the porous structure of activated carbon. This phenomena will require a longer contact time. Thus, when the contact time is longer, there will be more diffusion, so more methylene blue can adsorb onto the surface of activated carbon, causing the final concentration of methylene blue to be lower. This will result in greater percentage of dye removal.
Hi, I'm Laila Zarifah and I will explain the remaining parts of results and discussion. Next is to investigate the effect of pH level. The pH level of a dye solution was adjusted from 1 to 10 and showed different percentage of removal. We found that as pH increased, the percentage of removal increased until its breakpoint pH. And beyond that, will lead drops of the percentage of removal. The literature report that the absorbent surface is positively charged during the adsorption process at a low pH, which favors the adsorption of anionic pollutants. Since methylene blue is a cationic dye, the positive charge at the visible adsorption site competes with dye molecules, resulting in reduced dye adsorption. As a result, the maximum removal and lowest final concentration of methylene blue was observed at pH 5. Percentage of removal efficiency remains low in acidic and basic condition. The last part is to study the effect of mass of adsorbent. We found that by raising the mass of adsorbent, the percentage of methylene blue removed will improve. Considering increasing the mass of adsorbent beyond 0.6 gram of activated carbon did not result in a significant increase in adsorption rate. It may be due to an increase in adsorbent sites as well as more specific adsorbent surface available, which allows for greater dye adsorption at the given initial concentration. In general, the greater the mass of adsorbent, the greater the surface area of adsorption sites available. Hence, the higher percentage of removal efficiency of methylene blue.
Hi, my name is Michelle, and I'm going to present for the conclusion in this report. To summarize, we investigate the effect of contact time, pH, and the mass of the adsorbents towards the percentage of removal efficiency of methylene blue by using activated carbon. The best fit of the adsorption isotherms are determined for the adsorption behavior of activated carbon by plotting the Lammer isotherms and Frelich isotherms. So based on the comparison between both isotherms, we conclude that the Lammer isotherms is the best fit of the adsorption isotherms for the removal of methylene blue by activated carbon. This is because the R-square value of the Lammer isotherms, which is 0.9844, is higher and closest to 1 compared to the Frelich isotherms. The Lammer model has a higher consistency compared with the Frelich model, and the experiment results are more favorable and fit better to Lammer isotherms. Therefore, we can conclude that the adsorption of methylene blue on the activated carbon is a monolayer adsorption since the experiment fits better to the Lammer isotherms. All the objectives for this experiment were successfully achieved. With that, I end my presentation. Thank you.